Imagine living to 150, 200, even 250 years old. Sounds like science fiction, right? But what if I told you that scientists in Japan may have just cracked open a door to dramatically slow, possibly even reverse, elements of aging? In today's video, we're diving deep into how researchers at Osaka University uncovered a protein that acts like a youth switch inside our cells, and a companion drug that helps cells clean themselves out. Could this be the future of human longevity? Let's explore. Don't forget to hit like and subscribe, because we're about to unpack the science behind what's being hailed as one of the boldest anti-aging breakthroughs yet. First, let's ground ourselves in what aging actually is on a cellular level. Our bodies are made of trillions of cells, and over time they accumulate damage. Misfolded proteins, impaired energy systems, DNA wear and tear. One key culprit is the decline of proteostasis, the ability of cells to maintain healthy protein balance. Researchers define proteostasis as the sum of cellular machines that fold, refold, degrade, or recycle proteins. When it fails, damaged proteins build up, cells malfunction, and age-related diseases arise. Add to that the phenomenon of senescent cells, cells that have stopped dividing but don't die. They accumulate, secrete harmful signals, contribute to inflammation and tissue degeneration. Understanding and targeting them is a major frontier in longevity research. So when we talk slowing aging, we're talking about fixing these underlying problems, not just pills that make you look younger. Today's research is squarely in that deeper arena. Here's where things get really interesting. Scientists at Osaka University found that a protein called AP2A1, adapter protein complex 2, alpha-1 subunit, is significantly increased in senescent age cells. What they discovered: older fibroblast and epithelial cells show thickened stress fibers, part of the cell's internal skeleton, and elevated AP2A1 expression. When they knocked down AP2A1 in old cells, many hallmarks of aging were reversed. Cells shrank back towards younger size, structural markers improved. Conversely, when young cells were forced to overexpress AP2A1, they prematurely showed aging features. They also found that AP2 works closely with another protein, integrin beta-1, which helps cells adhere to their surrounding matrix. Together, they seem to allow senescent cells to anchor and maintain their enlarged size via stress fibers. AP2A1 behaves like a switch controlling cell state between young and old. If you can flip the switch, you might reverse aging at a cellular level, not just cosmetically, but functionally. Here's a useful analogy. Think of cells like old cars. Over time, they accumulate rust, broken parts, clunky systems. AP2A1 is like a lever that shifts the car into maintenance mode or retirement mode. Pulling the lever the right way lets the car resume smoother operation. The research suggests that reducing AP2A1 flips cells back into younger mode. This research is in cell cultures and early models. It's not yet showing human trials of live to 250 years. So yes, it's exciting, but not a guarantee. We'll talk more about that later. Parallel to the AP2A1 work, another major target in anti-aging research is enhancing the cell's cleanup service, the system that clears damaged proteins and structures. Two main mechanisms: the ubiquitin proteasome system, UPS, breaks down individual damaged proteins. Autophagy, the process by which cells engulf and recycle larger components like worn-out organelles. Researchers at Chungang University, South Korea, found that a small molecule drug called IU1, which inhibits the enzyme USP14, can activate both of those cleanup systems concurrently. IU1 increases proteasome activity and promotes autophagy in fruit flies and human cells. In fruit flies, IU1 treatment extended lifespan and improved muscle function. The mechanism appears proteasome-dependent but FOXO-independent, i.e., works through a different pathway than many conventional longevity genes. In plain language, it's like giving your cells trash collectors and recycling plants a major upgrade. They clear out the debris faster, restore cleaner internal conditions, which helps the cell function better for longer. Now, combine the two. One research line, AP2A1, targets the state switch of old versus young cell. The other, IU1, boosts the housekeeping within the cell to keep future damage at bay. The speculation: if you can both flip the switch and maintain internal health, you might meaningfully delay aging. There is no credible scientific evidence yet that IU1 or AP2A1 manipulation yields human lifespan of 250 years. The 250 years figure currently is speculative extrapolation in popular media. Now for the million-dollar question. Could humans really live to 200 plus years? Here's where we separate hope from hype. In cultured human cells and model organisms, manipulation of AP2A1 reversed age cell markers. IU1 improved age-associated decline in flies and enhanced proteostatic clearance in human cell lines. These are foundational steps, identifying targets, showing mechanistic proof of concept. Human clinical trials showing lifespan extension. Safety data over decades in humans for these interventions. Proof that one or two pathways alone can fully arrest or reverse aging. Aging is multifactorial. Telomeres, mitochondrial dysfunction, epigenetics, immune senescence, etc. Proof that flipping the switch produces a healthy extended lifespan. Living longer but disabled is not the goal. Thus, when you see headlines like Japanese scientists may extend humans to 250 years, treat them with caution. That number is more vision casting than fact. Why the 250-year figure surfaces? Because if you assume you slow aging significantly, say a doubling of health span, and push boundaries of cellular repair, then in theory the upper limit of lifespan rises. But this is speculative, and many other variables intervene. Disease, accidents, genetics, environment. What this could mean practically, short term, better therapies for age-related diseases, arthritis, Alzheimer's, heart disease. By targeting senescence and proteostasis. Improved health span, years lived healthy, rather than just lifespan. New biomarker targets like AP2A1 that might let us measure biological aging more accurately. I believe the most realistic outcome in the next 10 to 20 years is not humans routinely living to 250, but significant improvement in how we age. We may see treatments that delay onset of chronic diseases, reduce frailty, and add healthy years, maybe 10 to 20 extra quality years rather than doubling life. But the step from cell animal data to safe, effective human longevity interventions is still large. Also, there's a social ethical dimension. If lifespan increases, what happens to pensions, healthcare, work life, resource consumption? The science doesn't exist in a vacuum. All right, now I want you to think about this. If a drug came out that let you live, say, to 120 or 150 with excellent health, would you take it? Why or why not? Write in the comments below. Rather, pause the video now for 5 seconds and leave your thoughts. Here are 3 questions to consider as you follow this field. 1. Safety versus longevity trade-off. Do we accept possible long-term risks if the reward is extra decades? 2. Quality versus quantity. Is living to 200 meaningful if many years are spent in poor health? 3. Access and equity. Will such treatments be available only to the wealthy, creating a new health inequality divide? Human trials of AP2A1 inhibitors or modulators. Check clinicaltrials.gov for anything recruiting. Human safety efficacy data on IU1 or USP14 inhibitors. Biomarker tools that measure biological age, for example, the recent steroid metabolism model at Osaka University. Research into combinations of therapies, e.g. proteostasis plus mitochondrial repair plus senolytics, because aging is likely multifaceted. If you like deep science videos with clear analogies and big ideas, smash the subscribe button and hit the bell so you don't miss follow-ups in the longevity space. To sum up, researchers have identified a protein, AP2A1, which acts as a switch between young-like and old-like cell states. A companion line of research shows that IU1, a USP14 inhibitor, can boost cellular cleanup systems, proteasome plus autophagy, and extend lifespan in model organisms. Together, these suggest that we're getting closer to mechanistically addressing aging, not just cosmetic treatments. But we are not yet at the point where humans will live to 250 tomorrow. Major hurdles remain. The more likely near-future scenario, improvements in health span, disease delay, and incremental lifespan extension. Aging has always been humanity's only unavoidable shared enemy. But now, in laboratories, tiny molecular levers are being discovered that may shift the nature of our biological destiny. For those curious and excited about longevity, this is one of the most intriguing stories of our time. If you enjoyed this deep dive, please give it a thumbs up, share it with someone who loves future tech, and check the video description for links to the original studies and further reading. Thanks for watching.